Much. It's outstanding work. So the next presentation is from Olivier Commowick from INRIA. Will talk to us about MedInria, this important neuroimaging software for diffusion MRI.
There you go.
Great, thank you.
Okay, thank you. Why did this start? Okay, so I'm here on behalf of the MedInria team. So it's a team composed of 4 teams at INRIA. And I'm going to present you, well, yet another software for medical image processing, but with a nice focus on diffusion imaging, and I hope it will be helping some of you for your diffusion work. So I'm going to go over this using this roadmap. I'm going to present the MedInria overview, where we're coming from, where we're going, how it's architectured, then going on to some application examples, mainly relating to diffusion imaging and data management pre-processing, and finally going to some future directions and a quick summary of the presentation. So going on to MedInria. MedInria so it was created in the beginning by Pierre Fia at INRIA in 2007, and we started rewriting it completely from 2009, and with the goal in mind to transfer our research work in medical imaging at INRIA to our clinical, industrial, and our academic partners. So it's a joint effort of 4 INRIA teams, which are very complementary. It's Athena, directed by Rachid Dorish, Asclepios, directed by Nicolas Ayash, Parietal, directed by Bertrand Thirion, and VISAGES by Christian Barrio. And so we really have, because we want to transfer to all those people, we really have the philosophy to bridge the gap between the engineer point of view and the clinical-oriented point of view. Meaning that we want to have an easy-to-use application for research clinicians so that they don't need to have an engineer next to them to understand what the application is doing, how it's working. And we also want to have cutting-edge research because we are doing a lot of research in diffusion imaging, and we want to have this available to our partners to use these tools. So going a bit more into the technical details, MedInria is made principally of 2 main layers. The first layer is the core of the application, which contains the medical imaging related concepts like what is the data, what is the process, what it does. And then the second part of this first layer is the Qt-based interface. Well, the global shape, the global overall look of the interface. Then the second layer of the software is the plugins, and really the plugins are here to bring the functionalities. So there is really a clear separation between the interface of the software and the functionalities. And so the application gains from this because it's easily enriched by new functionalities. You just have to call a plugin to bring a new functionality. Then, in terms of interface, we work with workspaces. So I'm going to show you some examples just afterwards. But a workspace is either a group of functionalities that look the same, like segmentation or registration, or it's related to an application. And the one I'm going to present here today is diffusion imaging. So one of the interfaces that we have in terms of workspaces, just to show you that this is the visualization workspace we have, and there are always 3 parts in the software. Whoops. Okay. The first part on the left is the access to your patients, your files, everything you want to see. The second part is the views, where you can view your data, interact with it, play with it, see the results of your processing. And the last part is the tools, where you actually have all the parameters, the controls you can have on the views, controls on the parameters of your algorithms, things like this. So now that you know what a workspace is, we have 5 main workspaces in MedInria. Of course, today I won't have time to present all of them, so I'm going to skip some of them. But just to give you a quick overview, this is the visualized workspace that I already spoke about. You can view 3D data, 4D data, you can view fibers, you can view diffusion images, tensors, surfaces, well, whatever you can do with other softwares as well. Second workspace is the workspace for filtering, where you can apply filters to your images. So you have the input view, the output view, and you can run your algorithm and see what it does, what the result of it is. The third one is segmentation, so I'm not going to go very much into it today, but it allows you to define some regions of interest, to draw them manually, and then you can save them into your database and use them again in the other workspaces to, for example, to filter your tractography. Then next is registration. We have a lot of history in the labs that are implied in this software in registration. And so we are proposing a registration workspace to register Scala images right now, but there will be soon tensor images as well. Finally, the diffusion workspace, and I won't say much right now because I'm going to present it in a much larger focus just after. So these are the 5 workspaces of MedInria, and that concludes the overview of the software. So now going into some application examples. So I'm first going to present you some stuff about data management and pre-processing because it's often required to process your data. And then I'm going to go into diffusion imaging. Oops. So let's first speak about data access. It's not really visual, but we have an internal MedInria SQL database where you can view your patients, edit them, see what their age, some of their information. You can import a lot of various file formats in MedInria, formats supported by ITK, by VTK, some other more specific formats developed at INRIA as well. And you can also interact with external servers. So you can interrogate a PAX server, meaning that we are able to import DICOM using DCMTK. And you can also interrogate other sources. We have right now Shanwa, which is a data management system developed at INRIA, but you could also develop a plugin to interrogate an XNAT server. So this is the local database interface. I hope it's visible. You have a list of patients. You've got some controls to add patients, remove some of them. You can view the details of the patients. You can also drag and drop the patients. If you imported it without knowing its name, it's useful. So just really some stuff that you can use to manage your database. The second screenshot is the interface for PAX interrogation. And so here you have quite a few tabs here, but you have the list of patients that you have on your PAX system, a list of series that you can see. You can see also a preview of the series you're looking at. And then you select the images that you want to import, and you import them. They end up in the database of MedInria. Then going on to pre-processing, well, basically this is the filtering workspace. So there is a specific workspace for it, and you have an interface to choose and run a filter, to visualize your result, to save it if you want to keep it. You have several available features right now. You have basic filters, which are implementations of some stuff that are available in ITK, like Gaussian smoothing, median filtering, stuff like this. And you also have NLMIS denoising, which is a powerful denoising algorithm. And because it's really useful to denoise your images before processing them, we included it. So we have Gaussian noise support and Rishon noise, which is pretty much useful for DWI images. So this is again the interface, and this is an example of denoising performed by the NLMIS algorithm with Gaussian assumption. And on the left, you have the original image, on the right, the processed image. So that was pre-processing. Now going on to the really nicer part in terms of screenshots is diffusion. So we have, for the diffusion workspace, a set of user-friendly UI toolboxes that really have the goal to ease the configuration and the use of tractography. And so it provides a complete pipeline for DTI processing right now, from DWI brain masking, the DTI estimation from the DWI, tensor visualization, scalar field extraction, all brain fiber tracking. All of this has been proposed by Fia et al. In 2007 and is implemented in MedInria. And finally, you can view the fibers once you've done tractography, and you can filter them, bundle them, and once you have the bundles, you can also compute statistics on them. So I don't know if I will be able to start this video. Oh yes, thank you. So this is a video illustrating how the pipeline in DWI is working. So here you have a DWI that has been loaded, and you're just going to drop it into the tractography pipeline, and it's going to ask you for the gradients, so this is taking a bit of time, because you have to find the file. You can already see the toolbox that is going to come up next, which is the one to construct scalar maps from the diffusion tensors. And when you click on one of those buttons, it's going to estimate the tensors. If it's not done yet, it's going to mask the brain. And I don't know, why it's not moving anymore. And yes. Once you have done this, at the end, it's computing the scalar map and it's displaying it. Then, once this is done, you can go on the second part, which is the tractography. You can set a few parameters of the algorithm and then just run it once you're happy with it. And so, once this is done, once again, it's slow. Might be too big for the... Yes, this is going up. So once this is done, you can just run the algorithm and see the algorithm running. So this is the long part, so I've just shortened it, and now you have the fibers that are displayed onto the FA, and you can view them into the brain. So it's all brain tractography, so you get all those fibers, all these spaghetti plates, into your brain. So you can just view the fibers. The first one was local orientation, but you can also show different colors depending on the orientation or on FA values, for example. So now the last part of the tractography pipeline is the bundling part, which is controlled by this toolbox on the right. And so here the guy is doing some selection using this cropping box, and it's keeping only those fibers that are going through this cropping box. So it's one way of bundling the fibers. The other way is to open some regions of interest and to apply them so that you select only those fibers that go through the regions of interest. It's good that it paused, because there is also this thing. Once you have selected your fibers that you want to see, there is also a set of values that are computed from the fibers, and you have the mean variance, for example, of the fibers, the FA, ADC, some values like this that you can see. And so you can bundle using different regions of interest. You can have as many bundles as you want. So I think this is, yes, the end of the video. So that was the diffusion workspace in MedInria. Now, just to summarize and go on to future directions. So I hope I've shown you that we have an integrated interface for diffusion image processing. I hope it's an easy-to-use interface. You will tell me. It's plugin based, which is really nice in terms of bringing new functionalities. There is data management, pre-processing up till tractography. The software is multi-platform, is supported on 2 Linux distributions, macOS and Windows, and it's already available from med.inria.fr, and a new release is coming up in the next few weeks or even in the next few days. Now, what is coming up next? Well, somehow it's something we have already seen in other presentations. We have ODF processing and visualization that is on the shelf. It's very close to us. And what is very interesting is that it's bringing more information on crossing fibers, on splitting fibers, and also, in case of diseases, on their deterioration. So I'm talking about ODF because we have it almost done right now, but I'm also thinking about multi-compartment models, such as multi-tensor or the NODI model that we've seen just before. So this is a visualization of ODF models that we have in beta right now, and it's not available yet, but hopefully soon. The second point is tensor-based registration, which is once again bringing higher precision in terms of aligning the patients, and especially for the white matter. So we are going to bring that very soon as well. And finally, the last part we want to do very much is the pipeline construction interface, because in your studies, as has been said before, you want very often to do atlas construction, group comparison, one patient versus multiple controls comparison, and this requires automation. And to do this automation, you need to define your pipeline, build it, and then let it run on a set of data that you want to process. So we need to have a way to construct pipelines, and this is something we're working on. And well, one more thing is that MedInria is now open source. So the code is available from GitHub. So it's Qt-based, it's plugin-based, and it's really easy to extend the functionalities by adding new plugins. And so just a quick example, if you want to build a new plugin, you just have to create a canvas. We have some, well, very rough plugin generator right now, but it's on the way. You can adapt the toolbox for the parameters and connect it to your code, and basically you're done. If it fits in one of our workspaces, you don't have much more to do. And with that, I would like to thank you for your attention.